Assalamu alaikum and welcome back for our third and the final session.
Good to see you.
I take my pleasure to call upon our chairman, Dr. Mohammed Rukban, Vice President for Academic Affairs, to introduce our next speaker, Professor Mark Selvin, who is the Head of Genetics and Microbiology Department, United Kingdom, Manchester.
Bismillah ar-Rahman ar-Rahim. Alhamdulillah. Wassalatu wassalamu ala Rasulillah. First of all, I'd like to express my happiness and welcome to everybody in this auditorium. I would like also to welcome our guest speakers for accepting the invitation to be with us this morning and afternoon. And hopefully with this collaboration we will build up a trust relationship with the international figures and experts worldwide. I'm also feel happy to introduce one of my best friend, Professor Mark Selvin, whom I had chance to visit him in his office last year. Prof. Mark, he's one of the famous pathologists in the world actually, and his publication now is a well-known guideline for the field of cell pathology. In short, I think he have detailed CV, rich with many positions and publication achievements. We would like to congratulate him for this scientific achievement. Prof. Selvin, now he's the head of Genetics, Cell and Molecular Biology Department in Manchester Metropolitan University in the United Kingdom. He is a professor of cell pathology and acts as the standing chair in the cell pathology in the School of Health Sciences, Healthcare Sciences. Prof. Selvin also serves as the visiting chair in the clinical biomedicine at St. Paul Hospital in Barcelona, and he is playing and entire being a fellow of the Royal College of Pathologists and has been in this position since 2008. Prof. Selvin has recently been awarded the title of Eminent Scientist of the Year in the Cardiopathology by the International Research Promotions Council in 2011. He is a fellow of the Institute of Biomedical Sciences in the United Kingdom and recognized expert in the field of angiogenesis research. Prof. Selvin is also a member of panel expert reviewers of Wellcome Trust. He is also a reviewer in the ERA, NIT and Mid Research Council and at BPSRC. Actually, I don't know what is BPSRC. Maybe he can introduce it for us. Moreover, he is a fellow of numerous societies, including the European Stroke Association and American Heart Stroke Association. And to my heart is one of our collaborated scientists with the university. His input with the College of Allied Medical Sciences is welcomed by the university and thankful. Prof. Selvin, he will talk this part about nanotechnology, modulation of atherosclerosis and nano-targeted combinational stroke therapy. Prof. Selvin, you are welcome. Sorry.
Thank you very much. Hello? Okay. Thank you very much, Mr. Chairman, for your extremely kind comments. So, the previous two speakers have given a little bit of an introduction before starting. And I was thinking to move straight into the slides, and then I thought, no, let's introduce it. Why am I talking about modulation of atherosclerosis, for example? In health, in our life, we constantly strive to improve our health as we age, and basically to remain healthy for longer and longer. And this is what seems to have happened over the decades and the centuries. But we're reaching limitations to some extent, and it seems medicine has slowed down. The improvements, therapeutics have slowed down. One might argue that in 100 years, we might be able to cure cancer. So we may be able to cure the majority of types of cancer. In 100 years from now, maybe we have a cure or something which slows down or prevents development of Alzheimer's disease. various diseases which are very potent, and they quite often kill people in the prime of their life, not necessarily people near to the end of their expected lifespan. However, if we think about the future, and if we think about how we're going to the possibility that we can transplant organs into people, repair, replace, grow new organs, new parts of the body, cloning, maybe we can clone, so we have we have all of the elements that we need in order to revitalize a person as they get older. A couple of the biggest problems that we might have are dealing with a disease which is inherent in everybody, and which is development of atherosclerosis. Now, development of atherosclerosis begins when you're a child. There is some evidence to suggest that even there's damage to the wall of some of the major vessels before birth. So there is some type of issue or problem with the vascular system of the body which causes this. And as the atherosclerosis develops, you get plaque formation, and then this generally leads to ischemic stroke, or it may lead to myocardial infarction. Okay, so coronary arteries, carotid arteries. What can we do about this? At the moment, absolutely nothing. Okay, there is no ideas, particularly, for how to slow down or prevent the development of this disease. So whereas we can replace the heart, we are not very good at replacing coronary arteries or carotid arteries. Grafts, they are made from saphenous veins, for example. They're not suitable for the purpose. This is a serious limitation in the future of medical development. So when you get to the point that you're having high-grade atherosclerotic plaques which are liable to undergo thrombosis, let's say that this is in the carotid arteries, you have thrombosis and you get an ischemic stroke. What effective therapies have we got for treatment of ischemic stroke? The answer is very simple: zero. There are none. And in the last 20 years, there have been no new developments. So this is a very severe limitation. What is the problem? Our understanding of the disease? Is it possible that we can do something about it? And this is where nanotechnology may play a role. Not only nanotechnology, but nanotechnology for sure has applications in this field. And that's why I want to talk about specifically this area of health and disease. So, first part of the session, modulation of atherosclerosis using state-of-the-art nanotechnology. Okay, so in the 70s, Judah Folkman realized that a tumor cannot grow more than approximately the size of a pea without a vascular supply. So what the tumor does is it secretes molecules and then induces angiogenesis, blood vessels to grow out and from the normal body into the tumor, providing it with a blood supply. Seems to be that it was almost 25 years since then, before it has recently become accepted that the process of angiogenesis and vascular growth and vascular development is very closely linked to the way and the speed at which atherosclerotic plaques develop. So now it's totally 100% accepted that the process of angiogenesis, the growth of vessels within the arterial wall and within the plaque itself, is very, very much responsible for the plaque development, the instability of the plaque to a large degree. So whilst I'm not going to go into the pathophysiology of the development of atherosclerosis, it's worth it for anyone who's not, worked in this area particularly, to realize that right at the very beginning, you have vascular damage from the luminal surface caused by shear stress, for example. When you get macrophage build-up inside the arterial wall, lipid deposition, signaling, lots of cell signaling, activation, and you start to get a plaque forming, that plaque is like a tumor. It needs a blood supply. So without a blood supply, it will develop slowly, but in a stable fashion. With a blood supply, it takes in inflammation, it takes in microvessels that are unstable, and I will show you some examples of that in a minute. So big, big impact. You have an unstable plaque with microvessels within it which are liable to hemorrhage because they're not produced correctly by the body. This is causing an unstable plaque liable to undergo thrombosis. So angiogenesis, vascular system, is very critical to this development. I've put there 3 years nanotechnology. So between the time that it was realized that angiogenesis is accepted to be extremely important in this process, we already come up with a potential solution using nanotechnology. And this mainly, if you think about it, can be not for treatment necessarily, but delivery of therapeutics. So I'm focusing really on effective delivery of therapeutics, which is where the problem has been in medicine so far. So unfortunately, maybe the small screens, some of these pictures will be a little bit clearer. Because on the big screen, it's a little bit pixelated. And what we have here is an example. I'm going wrong way. Oops. Showing you all the slides now, taking the fun away. So here we have an artery. Normally, there is no intima. It's just a layer of endothelial cells. And in this case, we have a plaque developing. We have a large intimal area. We have the media, and we have the adventitia. And everybody tends to ignore the adventitia, which contains the vasa vasorum, the blood vessels which feed the actual artery itself. And normally these are ignored, but this is very, very, very important. We have some higher powered pictures, and we've done lots of studies to measure how many vasa vasorum there are in the adventitia in relation to where a plaque starts to develop. So remember, it's a three-dimensional system. The damage to the artery can be in any particular place within the artery, any part of the wall, thinking of it in 3 dimensions, not 2 dimensions, okay? And here is just a close-up of some of the vessels that are in there. These are functional vessels. They work there. They have pericyte coating, smooth muscle coating. They function. They don't leak. They're very good, yeah? They're very necessary. What is the relationship to vasa vasorum concentration? Okay, very, very interestingly, it has been shown plaques tend to develop where there are lower numbers of vasa vasorum in the adventitia. Not higher numbers, lower numbers. Probably because in that area there is a higher potential for hypoxia, a lack of oxygen supplying that part of the artery wall. And for that reason, when you start getting this activation of hypoxia-related signaling, this is causing then proliferation, which is what you don't want. Ideally, what you want here is quiescent cells. In the body, vessels do not proliferate unless there is disease process or during pregnancy. This is what we like in the body. We don't need the vessels to proliferate. When they proliferate, things start to go wrong. So, if we take... Whoa. I don't have to point it, do I? If we take a fragment of a plaque from an endarterectomy specimen, we see that it's heterogeneous. This is something we can't explain. Yeah? One small piece of it, of the same plaque from the same person, has lots of fragile microvessels, which you can see here, which are labeled with an antibody for active microvessels. And here, this is the area. The rest of it is quite stable. So we have an unstable area and a stable area. Why is that? It's very difficult to understand and explain why. This is very important because what we're trying to do is say, this is where your plaque will develop, and we want to protect you against getting this plaque. So we can't protect the whole vascular system, but we can say this is where the plaque will develop, and this is the area that's likely to be unstable, and that's the bit that maybe we can focus on. Because when we're looking at any targeted therapies, any use of nanotechnology, there are risks, toxicity. We have to be very, very accurate and focused and targeted in our therapy. This is absolutely for sure. One more picture, which again on the small screen should be very clear, and on this screen it is, not, is what the vessels look like. So I've just demonstrated to you that there are, in a plaque, there are vessels which are very, very thin-walled, liable to undergo hemorrhage. There are another vessel here which has a thicker wall and is a stable vessel. Okay, so these are very thin, and here, again, look at the shape of these microvessels. These are found within the plaques, and these are the ones which leak, hemorrhage, and are unstable, and which contribute significantly to the risk of thrombosis. So there are a number of notable studies that have investigated this in great detail. So what we have an hypothesis. Prevention, carotid or coronary, luminal endothelial cells. This is at the very early stages when damage occurs in children, for example. Right? Adventitial vasa vasorum, which are the vessels in the adventitia which become activated and then feed the growing plaque. Proliferation at known plaque development sites, so hotspots we call them, yeah? Using novel nanotargeted therapy might make a big difference. Will it work? That's the question. That's the hypothesis. Could it work? The hypothesis will slow down or prevent unstable plaque formation. The final level, if you already have plaques and they're unstable, can we stabilize them? Could we use some type of therapeutic method to release drugs which will stabilize an existing unstable plaque? Maybe we can as well. And there are studies which have shown, when you look at shear stress, using three-dimensional modeling, you can actually identify the regions which are at risk. The regions at risk are those where there's high shear stress, and those areas where normally the arteries are changing direction or bifurcating, so they become narrower. And there's like a much slower flow. There's more likely that platelets will get stuck to the side of the artery. So these things are very, very important. The knowledge to understand how we can say exactly in a three-dimensional format where this plaque is most likely to occur is very, very important to know. So, I like this picture. It's basically just demonstrating the importance of the angiogenic switch. So, normally in the body, vessels are quiescent. So, they don't look very happy because they're bored. They're not doing anything interesting. But this is what we like. So, you have a variety of anti-angiogenic proteins produced which are sufficient to block any possibility of angiogenesis. An example where this goes wrong is in proliferative diabetic retinopathy, where the androgenic balance changes. Maybe in diabetics the proteins become glycated, they don't function so well. The anti-angiogenic components, for example, opticin, they're not anti-angiogenic anymore. More pro-angiogenic molecules are produced, and you get diabetic retinopathy, which can lead to blindness, which is devastating. Okay? How can we maintain this anti-angiogenic switch in an off state? Very, very important question. What happens to that then? Okay. Have we got really strong evidence to suggest that if we block angiogenesis, that we can stop plaques forming? We haven't got fantastic models. Rodent models are not perfect. In humans, atherosclerosis takes 30 years. 40 years, 50 years to develop. We have ApoE mice. They develops in a few weeks, few months. However, sometimes we have to use these first of all as preclinical ideas, and then later we can use larger animals like the porcine model is quite good. Sheep is not so bad. So, basically, a couple of examples. This is even in 2006, so it's a few years old, where basically they've taken a molecule which binds only to active microvessels. So it won't bind normal quiescent vessels, it's not going to damage anything, it doesn't bind to other cells, it's targeted. It's a targeted delivery mechanism. You have an imaging agent, gadolinium, so it's a contrast agent, so you can visualize this with, for example, MRI, and it's linked to a carbon nanoparticle, basically. This also then is connected to an anti-angiogenic drug called fumagillin, and they used, in this case, a rabbit aorta. So what they did was they had controls, and then they had this nanoparticle, which you can see by contrast imaging MRI. They injected it, and they, over a period of time, looked at the plaque development. What did they find? Just by having this strong anti-angiogenic agent, the plaque didn't form, virtually. There was no development of the plaque, suggesting that maybe if you inhibit angiogenesis, you could slow down the formation of plaques. So we'll move to the next one. Another example, treatment of atherogenic mice with plasminogen activator inhibitor, truncated one, reduced vasovassorum proliferation. So they've proven by histology that it reduced the vasovassorum proliferation and plaque growth in mice. And they went into some detail looking at the mechanism of how they did this. And the pictures are showing quite clearly, almost the plaque is not forming. What we do know is that when we transfer knowledge from animal models to humans, it doesn't usually work out quite the same. They're not usually as effective because the process is more complex. And finally, a different approach where basically a group augmented, increased, they'd injected in microspheres a growth factor which is pro-angiogenic. And in this case, this caused the vasovassorum to proliferate and the plaque developed in this region, this specific region. So there's two types of evidence, both very strong, suggesting that the angiogenesis, if we can modulate angiogenesis, maybe we can significantly slow down the development of the process. How are we going to do this? We have our drugs, we have maybe a system, a lentiviral system, where we could insert DNA into specific targeted cells that we want to secrete particular proteins that are anti-angiogenic, maintaining a microenvironment. The microenvironment, that's all we want, against angiogenesis. So this is one idea and possibility. Always a risk when you insert something into the DNA, a little bit of a risk of what will be the long-term effects of this. So we have some options that we mentioned here and some of the anti-angiogenic agents which are naturally occurring, so they themselves will not cause any damage to the microenvironment. Then we think about specifically the nanotechnology approach. How are we going to use nanotechnology within this? And we have various systems, different types of nanoparticles. Oromag, produced by a nanotechnology company in Barcelona, which they've tested in great detail, and they have been able to link various antibodies and sort of for example, HIF-1 alpha sensitive linkers to these nanoparticles. And basically, the system that you would have is you would have your Oromag, it's called. It's a gold super paramagnetic nanoparticle, which is quite important to know. We link it to the lentiviral vector. We have to have a therapeutic, a drug, or a mixture of drugs. Okay, more than one would be much more effective. And then how do we target it? With the integrin, for example. It's very highly specific for those vessels, just that group of vessels. And it is very, very important that we just pick, on a three-dimensional level, if you're looking at the vessel, which I'll show you a picture in a minute, one region, not even the whole circumference of the vessel at any one point. It's very important to be able to discriminate where exactly you're going to target. So, you need to do some in vitro studies, ex vivo. So you have a rat aortic ring. How are you going to get this therapeutic that you've designed to go to the place where you need it? Well, if you insert a catheter, for example, drug-eluting catheters are quite effective. It has been shown that even in the larger arteries, a diffusion process will allow small nanoparticles to diffuse from the intima through the medium and even to the adventitia. So maybe you can do it just with this targeted integrin, and it will diffuse through using a catheter-based delivery system. In some arteries, it may be more effective to use magnets. So remember, the nanoparticles are super paramagnetic. This means that you can actually apply a magnet, as is shown there, and actually you can move the particles through the tissue with the magnets and keep them exactly where you want them. This is quite feasible for carotid artery. For coronary arteries, it's a little bit tricky, maybe, but not impossible. So this is one idea for how we would employ magnetic nanoparticles. The other thing is, do we want to use 20 nanometer size gold nanoparticles? We sort of know what happens to them when they go inside the cells, and I have one slide later to show you that. Or is there a better alternative? I believe there is a better alternative. Nanoparticles are now accepted for use in humans for diagnostic purposes. For example, in imaging after stroke, you can use iron oxide nanoparticles at about 5 to 10 microns. Been demonstrated as a contrast agent. They don't cause almost any damage whatsoever to the brain. And they're also super paramagnetic. So we could consider using them as part of a magnetic delivery system for delivering our therapeutics. So another picture basically showing how you use a colorimetric map, what's called time average wall shear stress, or TAUS, basically, or OSI, in order to look at the hot spots within the region of the artery. Usually at the bifurcation site, we're concentrating on the carotid artery, of course, here, because we're looking at stroke. And we can say, look, this is the area, this is where the plaque is 95% likely to develop. This is where we want to target the vasa vasorum. We don't need to target all the way around the vessel. Why is that important? Because if you do three-dimensional computerized tomography, and you look at the structure of the arteries and the vasa vasorum, you can visualize them in 3D. I put this little picture on the end because somebody asked me in London at a meeting when we were discussing this as a possible therapeutic, and they said, can you tell us that if you inject your anti-angiogenic agents to the vasa vasorum of the vessels, what happens if you still get plaques forming in the same area? Can you, where are your collaterals going to come from? Are you going to get any sprouting collaterals, which usually, can save people's lives?
Yes,
it's very, very important, these collaterals forming. And if your microenvironment is purely anti-angiogenic, they won't form. So the answer is basically specificity to one area, not all around the circumference. It's very, very critical to be able to target exactly the vasa vasorum that you need to target, for example, in this.
I will call you back.
And so this is just another example how we would put the magnets, you put the catheter inside, the region that you localize, and of course, if you're going to, however, whether you're using magnets or whether you're just going to use diffusion, you would not coat completely the catheter with your viral package. You would only coat one region around the circumference of it. This is just the slide I wanted to show you about toxicity of nanoparticles. We have to be very, very careful when we're introducing nanoparticles into the brain particularly, but into any part of the body. How are they going to enter the cell? Are they going to stay in the cell? Are they going to be released by exocytosis? They have to be non-cytotoxic, of course non-inflammatory. Imagine you put something in which activates NF-kappa B and basically stimulates inflammation. This is it's a disaster, basically. So we have to characterize them very, very carefully. This is how they're internalized and they're removed by exocytosis. Just a little picture to show what they look like when inside the cell. And this, you'll see, is important later. You can put this small nanoparticles inside the cell without causing any damage and use them to move the cells around. It's quite interesting. So this is a prototype. We call it Achilles heel, and that's how it would be designed. We would actually probably not use the gold nano system, which is already available, but we would use this just as a prototype because it's already manufactured. We have our anti-angiogenic agents. We have our nanotechnology, and we have our targeting system. And that's basically what we designed already in order to try to target specific areas of a carotid artery near to the bifurcation point of the internal-external. And this is what we're using in an animal model to see if it prevents development of these plaques. A little summary. Consideration of 3 phases. Can we inhibit intimal endothelial cell activation in children or young adults? Probably by a drug circulating or by insertion in those hot spots only of something which prevents those intimal endothelial cells from becoming activated. Because when they become activated, they express adhesion molecules, the gap junctions get wider, they become permeable. That's how the macrophages get in, secretion of pro-inflammatory factors, and that's where your plaque starts, basically. Can we stop that? Can we slow it down? That will then slow down the next process, part of the process. Can we make little microenvironments at the hotspots, preventing the vasa vasorum from activation, and also improving the stability of any plaques that may develop, because they will not contain angiogenic vessels. If we have them already, can we stabilize existing unstable vessels? Again, by specific application of this anti-angiogenic molecules, stabilizing molecules, molecules which cause vessels that may exist to get pericyte coats, to function correctly. 3 phases, 3 possibilities at any stage of development of the disease. That's the first part finished. How long do I have approximately?
I think we have 20 more minutes.
20 minutes.
Yeah.
I can get through this. Yeah. This is no problem. Okay, so we failed to do this, and we have a stroke. We have an ischemic stroke. Okay, what can we do about this? Can add recannulate TPA, try to get some blood back into the brain. What else can we do? Very much, not very much. Yeah? This is what happens after stroke. It's not very simple. It's a very complex process. So, we have sometimes transient ischemic attacks, warning signs. We have silent attacks, ischemic attacks, which we're not aware of. This causes damage to the brain as well. Basically, over a time course, we can see different pathophysiological elements of the development of the stroke. And cytotoxicity, increased inflammation. Then you have these are the worst things. These are the things which cause all the death, cell death, very early on in stroke. The first 24 hours. After 3-4 days, inflammation subsides naturally. But it's too late, because you have an infarcted region which may be, quite large. It's too late to do anything about it then. So we need to act here. But if we're going to have an effective therapy, we need it needs to be combinational therapy, okay? We have hyperacute phase, acute phase, early remodeling, and chronic remodeling, which can go on for months and years. So, you can't see the top slide, doesn't matter. What are the key events? Speed of reaching a stroke victim, okay? The person is in hospital, perfect. person is at home, you miss the hyperacute phase. You miss part of the acute phase. It's very, very difficult after this point. Effective thrombolysis is very, very important. And then, if we can do that, we can consider applying our therapeutics through the same way, okay? Through the arteries, the arterial system. But there is a clinical puzzle. And why have there been no successful drugs used to treat stroke on the market in the last couple of decades? Question which was tried to be answered by this group here recently. Do stroke models model stroke? And of course they don't do it very effectively. And we did some work recently, a few years ago, where we took samples of brain tissue for patients who had a stroke from different times, a few days, a week, 10 days, 4 weeks, and we did gene microarray analysis. And then we did the same thing with the rodent model, which is used most of all. We took the same time points going all the way up to 4 weeks. And we looked at the genes that were expressed at different times. The majority of the genes are expressed very early after stroke. This is the problem. However, half of the genes that were expressed were different in the mouse model than they were in the human. So the pathophysiology is completely different. That's why, part of the reason why the models don't work very well. And I don't think you can see this very clearly unless you look on these screens, but they're basically just showing the times and the number of genes above the lines which are increased, and also reduced, of course, over time. They're quite different. And this picture, this is why I wanted the lights a little bit low. If you really want to see it, you have to look at the screens. Basically, this is an example. We spent 5 years looking at a drug, which is called CDK5, the protein, and the drug is CDK5 inhibitory peptide. After stroke, you get calpains produced in high quantities. What the calpains do, they hyperactivate that protein CDK5. So basically, that induced that in the neurons, it activates a cell death pathway, so apoptosis. That's the end of the neuron. You can't do anything about that. So we investigated its signaling and what it does in different cells. Apart from this, which is devastating, what it does when it's activated normally and correctly is it supports remodeling. It supports angiogenesis. So I was thinking, oh, wait a minute, with the drug, we can protect neurons, maybe, if we stop hyperactivation of this protein after stroke. It's the reason for 80% of cell death after stroke. But we can also improve remodeling with the same drug. Multiple action. This could be really interesting. Yeah? So here, it's just to show you one of 1,000 experiments, which was absolutely fascinating, where we demonstrated when you played the brain endothelial cells out, you have an actin cytoskeleton produced, and then you have a delivery of the substrate for CDK5 moving down the actin fibers to the tips, where it binds CDK5 and activates the cell. It's absolutely fascinating. We have real-time images of this happening as well. And here, when it reaches the edge of a moving cell, it binds with integrins and tallin. And together, they cause movement of that cell, motility, which is, in the case of endothelial cells, the key component of revascularization, vascularization, and angiogenesis. So I think I'll ignore that one. How are we going to use such a drug? How are we going to get it to stroke, to the area that we need to get it to, quickly as possible, so it can be effective and protect against the effects of excitotoxic neuronal death and inflammatory-induced infarct increase in size? And this stems from an idea, I'll go back to this, whereby in 2013 we have I think this has run out now, so we have an idea of basically using a nanotechnology where you make pH-sensitive particles. Okay, so you can manipulate the particles to become pH-sensitive. Now, in hypoxia, pH drops. Okay, so these particles are used in tumors, and they basically home to the the lower pH region, which is where you want them in the tumour, and they're killing the tumour effectively. So I was thinking, well, this is the same in stroke. It's exactly the same in stroke. Can we use them? Maybe we can. So what have we got? I think did I miss? Yeah, I put there at the top hyperacute stage 1. This is like up to 1 hour or 2 hours. If you want to introduce a therapeutic at this point, it has to be very quick. So it has to be a nanoparticle with some delivery agent on it, a drug released like a liposome, released effectively to the area of the stroke immediately. Okay? It's very difficult to do that. We haven't thought exactly how we're going to do that, but we can use the idea of pH sensitivity. But in the acute phase, we want to stop inflammation. It's absolutely critical to stop inflammation. What has been found recently is that we have patient-derived omentum mesenchymal stem cells, which have fantastic properties. And it's only published, there's only 2 papers published so far from collaborators that we have in Italy. And they demonstrated that you can load drugs into the stem cells. So we have these fantastic stem cells, and these stem cells can be anti-angiogenic or pro-angiogenic. You can change the phenotype of the cell. So in the inflammatory phase, the acute phase, we don't want inflammation. We don't want angiogenesis. We want a stable vascular system. We want to maintain the integrity of the blood-brain barrier, for example. So, stem cells, they also secrete lots of very useful components, and they can carry a drug. They can carry the drug and release it over a time course of 3 days. So, whereas an injection is one time only, if you can get these stem cells to the area of the stroke and keep them there, they will release a drug for 3 days, which is exactly the period of time of inflammation before it subsides. It's absolutely perfect. So this is what we're looking at now. We have to consider which drugs to put in. I suggested one already. There is not the only one. Interleukin-1 receptor alpha. We work with Nancy Rothwell, who's the Vice Chancellor of the University of Manchester. They've been working on this drug, interleukin-1 receptor alpha, for 25 years. It's extremely effective at blocking inflammation. So we will combine them. We will put them together in stem cells, 1 million of one, 1 million of another. The only problem we've got is how do we get the cells to the stroke area, the ischemic area, and keep them there? Because you have to keep them there, otherwise the body will remove them. They will go into the circulation, which is what happens when people have tried stem cell therapy. It doesn't work. It's not effective in stroke. So how do we hold them there? We put these superparametric ferrous oxide iron nanoparticles, which are used in diagnostics. And they're very, very small. They do no damage to the cells whatsoever. We load them into the stem cells, and then we can move the stem cells around with magnets. And a little example there on the bottom is levitating cells, which have nanoparticles inside them being held in a 3D environment by magnets. You can do anything you want with them. We have been doing this. We have been moving magnetized cells within the vasculature system of the brain in an animal model. So it works. What you can do there is then bring them to the place where you want them. You keep the magnets externally, holding them in position. And then the stem cells will release the payload over a period of days. And it seems that it might work. Yeah? So that's basically what I've described there. And this is just an example to show how we can manipulate the mesenchymal stem cells. So at this point we want anti-angiogenic stem cells. But later, when we will have remodeling, we provide a new set of stem cells with different drugs, and these are pro-angiogenic stem cells, and they're releasing lots of very useful factors, and also they can differentiate themselves into useful cells, into new vasculature, for example. A little bit of information about the signaling pathways and how we do it. Weeks and months later, we basically we have to consider, other types of approach to maintain improvement. But should the first 3 phases be effective, This would be much easier. But we do have to consider that when you have any type of stroke, you increase your risk of getting dementia, particularly vascular dementia, 5 to 10-fold. So we have to consider, how this approach will, it maybe, therefore, reduce the risk of going on to develop vascular dementia as we get older. Now, if I have 5 minutes, I'm going to skim through these just for a little bit of thought. We have this problem, if we want to live longer, maybe we have problems because we can't transplant a brain, can we? We are who we are, and I think it's a very difficult thing to consider. If you want, there was some evidence that Walt Disney, many years ago, froze himself down, hoping that in the future, technology would allow him to be brought back to life, to be repaired. But if your brain is really telling you who you are as a person, your memories and everything. Can we do anything? I mean, do we understand this sufficiently? If you clone someone, it's a different person. Of course it is. It's not you. Yeah? However, recently they grew part of a brain in vitro, in the laboratory. It had some synapses, some functional synapses. So we have this issue. Well, yeah, we can sort of maybe start to think about creating a little bit of brain tissue that functions. I just mentioned if we clone a person and reinstall all their living memories into a different brain, is that the same person or is it a different person? I don't know what the answer is. I guess it's not. And do they become something like immortal? Nanotechnology. What we've done is we've taken neurons, human hippocampal neurons, and you see if in these pictures on the right hand side, we use carbon nanotubes with chemical gradients, and we've actually made axonal connections between them, going on a microelectronic chip. Okay? Why are we doing this? Because we have a binary mathematical system like this, which is able to interpret not only synaptic firing, so it actually can calculate. You can make neurons add up. 1 + 1 is 2. Yeah? We can make them add up. Yeah? What else can we do? There's a flip-flop mechanism. What this means is that we can actually teach the neurons. They can learn something, a memory, and then they can repeat this cycle. So we can actually give them a memory in vitro. This is very scary. Imagine this in a 3D matrix, something outside the body which holds memory, a biological machine that holds memory. We can then place this in a computer because we have the mathematical hardware. Can we then reintroduce this back into the brain of something? And will it remember? So can we move memories in and out of something? I think it's a little bit frightening, really, but this is what we're doing. This is the microelectronic chip. These are the neurons firing. And that's my guess as to what would be the future if we continue down this road. 2013, we have a biological computer. 2023, we have what somebody might call a Genesis cube. Animal testing, inserting memories in and out of the brains of animals. 2113, human brain memory download into a computer. So Walt Disney didn't have any idea about this when he was frozen down, but in the future, somebody with lots of money, could they actually download all their memories onto this computer system using this software, and then in the future try to come back as the same person by reinserting all of their memories into a clone, into the brain of their clone? I don't know. I doubt it, but there we go. Hope not. Thank you very much. Any questions?
Yeah, many thanks, Prof. Slevin, for this interesting talk. and especially the last slide. It's very interesting, really, and create a lot of thought about the future of the mutations and coining and movement of the science toward the changing, initiating the new ideas to create a new brain. But it is really interesting to thought about what he call it as the biological computers. Now they are growing up. Anyway, we have few minutes for questions. If you have any questions or comments, please feel free. I think we have also a chance for the ladies to comment. Let us start from here. If anybody want to talk, yeah. Dr. Yunus. Okay, yeah, take the mic.
Thank you for your excellent lecture. My question is, instead of a gene, I mean, you are using a lentivirus, instead of a gene, can we use anti-angiogenetic agent to be delivered there using the nanotechnology?
So if I'm getting the question right, can we use a... so basically the idea was to use a lentivirus to insert into this lentivirus a gene sequence for a peptide or a protein.
Right.
Then basically when that virus inserts itself into the cell, so it will be specifically directed into one type of cell, that the sequence then goes, the lentivirus inserts itself into your own DNA, and then you start to produce basically increased amounts of the gene, increased amounts of the protein, which is secreted, and
so that's why not use the same required protein, which is anti-angiogenic agent, directly introduced to the location?
Because the problem is that, for example, in atherosclerosis, this process lasts for years and years, so you need something which is going to be effective forever, for your whole life. With stroke, for sure, we do not want to use that technology. For stroke, we probably would use something like mesenchymal stem cell to deliver the drugs directly. But in atherosclerosis, if you can actually just create this microenvironment which just doesn't allow any factors which may be trying to change the angiogenic switch to make it pro-angiogenic. We have a picture where we've seen in a coronary artery a track of an endothelial cell from the vasa vasorum going through the media into a plaque and forming a vessel. It's absolutely incredible. They actually are doing this, and this is what is forming making the plaque, allowing it to grow bigger and making it unstable. Whereas if that microenvironment was kept anti-angiogenic by the constant secretion of these proteins, this will never happen. This cannot happen.
So what is the adverse effect of this anti-angiogenic sequences is inserted, which is expressing the anti-angiogenic agent? What would be the adverse effect?
So there would be no adverse effect because they would be introduced into, let's say, the adventitia of an artery. Nothing is happening in the adventitia of the artery. Blood is flowing through the existing vessels. It won't affect the existing vessels. So it's only going to stop the active process of new angiogenesis, which is not supposed to happen. We don't want this to happen inside the body.
Thank you.
Okay. If you have any more question, yeah, please. Professor Wahid there.
Assalamu alaikum. Good afternoon, Dr. Slavit. Nice explanation. I just want to ask you one thing about your modulation finding in your school of thought regarding about the role of vasovasorum you have explained. You have just explained about some mechanism in the diabetic retinopathy. What we understood is about the role of this complication of retinopathy in diabetic is mainly because of the non-enzymatic pathway of the coagulation of protein. Right. Now, what is your school of thoughts? Because this is the most complicating situation where the management is quite difficult, and you are just trying to highlight something on the nanotechnology also. Yeah. And what is your school of thoughts in that? Yeah,
this is it is very difficult. The proliferative diabetic retinopathy. What we've done is there's a group of collaborators who've been looking at the changes which occur, which are maybe key in determining this switch. And there's one protein which is very interesting because during embryological development, it becomes expressed at a certain point, and at the same time you have vascular regression inside the eye. And so when you look at this, we took this protein, opticin, and we investigated it in great detail. And what we found was that in all the in vivo models, so the models of diabetic retinopathy, like the rabbit, for example, that when you apply opticin or an adenovirus containing opticin, which secretes opticin in the locations that you want it to, in the vitreous, for example, it prevented the development of proliferative diabetes. So it prevented blood vessels, which are again formed incorrectly, and they hemorrhage, don't they? So it actually is significantly, I mean, almost completely prevented this development. And what we also did, we found out that if you apply levels of glucose, for example, to this, high levels, you glycate the molecule. So you can glycate, when you glycate opticin, which is what you find in the diabetic eye, it's glycated, because in a diabetic patient, it loses its anti-angiogenic effect. So we believe that apart from the increases in some growth factors like VEGF, the opticin is holding the balance. And when it becomes glycated, it doesn't function anymore. So the balance is shifted. The question is what can we do about this? I'm not sure. I have to think about this. But for sure, this is very important.
Yeah, interesting.
Thank you.
Okay, if you allow, just want to take a few questions from ladies, if any want to participate. Any question? Okay, please. Can you bring the mic, please? Anybody there? Okay.
Yeah. I have one question, please, about the cost of because the atherosclerosis has 5 stages. If depending upon the average cost of the nanotechnology in case of atherosclerosis, roughly I want to know the cost of in different stages.
Yeah, you talk about the cost in different stages. Is this a question?
Please.
Yeah.
Wow. Yeah, this will be very difficult to answer. I would guess that in the first stage would be simply a case of thinking about drugs which would protect the endothelial cells. This would be something that's feasible, that wouldn't be very expensive, and there are things, naturally occurring substances like aged garlic extract, things which protect, against the sort of like damage and the oxidative stress and things like this at the early stages. But still, the most effective thing would be to be able to introduce a little gene sequences into the cells. The cost, we have a National Health Service in the UK, and I wouldn't guess that they would agree to some type of therapy. I would guess it would be very, very expensive. The only thing you have to consider is, if you look at somebody's genetics, you know from family history, you know from looking at you can do a whole genome sequencing. We know what their risks are of developing certain diseases of an individual person. So if my family history says to me that, it's very, very likely when I get to 65, my arteries will be blocked and I'm likely to have a heart attack or a stroke, it could be that selectivity, it means that there's a possibility I could have some type of therapy in the future. But it wouldn't be something that everybody would even need, because there are people who live 90 years old and they have, just basically a sort of hyperplasia of the intima, which is stable. And that's fine. That's not going to affect even your quality of life. It's not going to be the thing that kills you. So it would be very selective therapy, but it would be expensive,
Yes.
I guess.
And it's also expected that with the growing of the science and the use of such technologies, the cost will be reduced down.
Then it will start
with
to
the use
reduce.
alternatives.
Yeah, for sure.
But initially, to start with these innovative ideas, I think it's something need to be considered. Please.
First of all, I would like to thank you for this very informative lecture. Currently in medical in practice, stroke is primarily, primary prevention for stroke is, antiplatelets together with statins, and controlling the main cause of stroke, potential stroke. So, do you think that still with the use of these medications for primary prevention of stroke, still there will be potential changes in blood vessels, and which later could lead to a stroke because of the changes, as you mentioned in one of your slides, that the causes changes in vessels, not because of the other theories, or together maybe.
Yeah, I think, of course, there's lots of evidence that after stroke, you do get a lot of remodeling. The body tries to remodel the brain, and that includes inducing angiogenesis. But in this case, the angiogenesis is not very effective. We did like vascular casting of animals and things, and we looked at the way that the new vessels develop. And so you have this issue that the remodeling and how effective it is in terms of the new vessel development, how effective those vessels are at providing the nutrients and the oxygen to the damaged area is very limited. So there are drugs which you can give maybe to people who've had a small stroke, a transient attack, which would prepare basically the brain. So like citicoline, for example, things which are totally safe, statins to some extent, which have been one or two types of statins have been shown to have evidence of plaque regression in atherosclerosis, maybe increasing plaque stability. So there are those drugs which have those type of benefits. And of course the basic things, you can talk about aspirin and stuff which reduce the risk of a subsequent heart attack, for example, myocardial infarction. But they're, of course they're not that effective, because those people who have a stroke usually have a second stroke, which is usually more severe. We have also the problem at this stage of how you have plaques within a system, and how stable are those plaques. I'm thinking 90% really is capturing it before you reach this stage. And when you reach this stage, it is very, very difficult. And the idea to stabilize the plaque, so to basically find out exactly which areas of the plaques need to be stabilized, which have these inflammation and microvessels. Can we then stabilize those as an alternative type of therapy, as well as giving them conventional therapies, which basically protect the system against the thrombosis and the clotting and the platelet aggregation and things like this, which clearly are very, very important as well.
Yeah. Yes, please. A lot of hands now. Just make it short and short answers too, just to accommodate all of these, please.
Okay, thanks a lot for the wonderful lecture, sir. I want to know about that. You had talked about the combinational therapy of using the anti-apoptotic agent, that is CIP, to block the cyclin-dependent K5 kinase gene, and at the same time you are blocking the anti-inflammatory agent, because you don't want the initial inflammation within the 3 to 5 day window. For that CDK5 blockage, you had used CIP peptide, yours. Fine. Yeah. Have you looked upon the other anti-apoptotic genes like BAX and BAF and APAF, all those? And at the same time, you are using anti-inflammatory agent. And in the slide, you had shown that IL-1 beta, which you had looked upon. But there are many other anti-inflammatory, pro-inflammatory genes or pro-inflammatory cytokines like IL-6, TNF-alpha, IL-1, and I want to know more about this thing.
Yeah, so I'll try to answer that very shortly, very quickly.
Yeah.
with the CYP peptide, it's we want to look for something which is at the top of a pathway, so it's basically more effective. So wherever you tried in any disease, where you say, okay, we can block MAP kinase, for example, you can't block MAP kinase in a cell because you prevent all the naturally occurring signaling. So we need something that's right at the top, and which, for example, in the case of the CYP, CDK5, the calpains cause 90% of the damage, and they're produced in massive quantities. So we can't inhibit calpains. It's impossible. There's many of them, and they do lots of different things. Some of them are good things. But what we can do is say, look, we can prevent them hyperactivating this with this very specific peptide. So it seems to me that would be more effective than any other. But in combination, because and again, the interleukin, the group that we work with, they've studied interleukin-1 beta, interleukin-1 receptor alpha, the other interleukins, and they found that this one is by far the most effective. And I'm not sure exactly how that signaling pathway works, but of course it's right at the top. I think that's what it is.
Okay.
Thank you very much. As we know, atherosclerosis has many risk factors. You believe in the future we will help our patients by this new technology while we are using the risk factors like smoking and obesity and so on? Thank you very much.
Yeah, I suppose I would probably say, and it might not go down very well, is that if you smoke, you wouldn't be eligible for this therapy, which is maybe, wouldn't be the case. But for sure, smoking is one of the strongest risk factors for development. We know what the others are. If you can control high blood pressure, this is good. But the vascular system in the human is very badly designed. This is the problem. If we were 25 feet tall, and all of these branches and turns were more smooth and soft, we would have much less possibility of atherosclerosis. It's a mistake in the design of the body. Yes.
Yeah, I think Dr. Asr is adding this as a joke. Definitely nobody will believe that we should take this innovation in technology just to have the take it. Okay, any question from the ladies' side? Okay, if not, then I think it is really an interesting lecture, and I can say at the end of this nice presentation that I learned a lot from the short presentation of Professor Slevin. Maybe the most important point in my view that he opened our eyes toward the thinking about, the sense of the research and how researchers should appear. And this important message to our staff, and especially for our junior student and the staff, that presence of such sciences in our university encourage us to think about this way of scientific approach. Yeah, to go to this depth in management and research. Really, it is amazing in my thinking. Thank you, Professor Slevin, for this interesting talk. Thank you for you all for the attendance and interaction. I think we are waiting for the last session of today's meeting. Thank you very much. And in a few minutes, they will start the last sermon. Thank you, Prof.
بسم الله الرحمن الرحيم. الحمد لله القوي المتين القاهر الظاهر الملك الحق المبين، لا يخفى على سمعه خفي الأنين، ولا يغيب عن بصره حركات الجنين. ذل لكبريائه جبابرة السلاطين، وقضى القضاء وهو أحكم الحاكمين. وأصلي وأسلم على سيد الأولين والآخرين نبينا محمد وعلى آله وصحبه أجمعين. سعادة وكيل الجامعة للدراسات العليا والبحث العلمي الدكتور محمد بن عبد الله الشايع، سعادة وكيل الجامعة للشؤون التعليمية الأستاذ الدكتور محمد بن عثمان الركبان، أصحاب السعادة عمداء الكليات والعمادات المساندة، سعادة الأستاذ الدكتور أوليفر بوناوت، سعادة الأستاذ الدكتور مارك زيلفن، سعادة الدكتور بخيت بن عوض الرشيدي، مدراء الإدارات، أعضاء هيئة التدريس، ضيوفنا الكرام، السلام عليكم ورحمة الله وبركاته. بقلوب ملؤها المحبة وأفئدة تنبض بالمودة وكلمات تبحث عن روح الأخوة، نقول لكم أهلا وسهلا ومرحبا بكم. الحياة مليئة بالعلوم التي أنتجتها عقول البشر، والتي وهبها الله لكل تلك العقول من إلهامات وعلوم وحكم. واليوم تقيم كلية العلوم الطبية التطبيقية بجامعة المجمعة بالتعاون مع شركة وادي السيليكا لتقنية النانو ورشة عمل بعنوان تقنية النانو وتطبيقاتها في المجالات الطبية. وخير بداية لكل بداية آيات بينات من الذكر الحكيم يتلوها الطالب عبد الله منور المطيري.
أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. قل لا أقول لكم عندي خزائن الله ولا أعلم الغيب ولا أقول لكم إني ملك إن أتبع إلا ما يوحى إلي. قل هل يستوي الأعمى والبصير أفلا تتفكرون. وأنذر به الذين يخافون أن يحشروا إلى ربهم ليس لهم من دونه ولي ولا شفيع لعلهم يتقون. ولا تطرد الذين يدعون ربهم بالغداة والعشي يريدون وجهه ما عليك من حسابهم من شيء وما من حسابك عليهم من شيء فتطردهم فتكون من الظالمين. وكذلك فتنا بعضهم ببعض ليقولوا أهؤلاء من الله عليهم من بيننا أليس الله بأعلم بالشاكرين. وإذ جاءك الذين يؤمنون بآياتنا فقل سلام عليكم كتب ربكم على نفسه الرحمة أنه من عمل منكم سوءا بجهالة ثم تاب من بعده وأصلح فأنه غفور رحيم. وكذلك نفصل الآيات ولتستبين سبيل المجرمين. قل إني نهيت أن أعبد الذين تدعون من دون الله قل لا أتبع أهواءكم قد ضللت إذا وما أنا من المهتدين. قل إني على بينة من ربي وكذبتم به ما عندي ما تستعجلون به إن الحكم إلا لله يقص الحق وهو خير الفاصلين. قل لو أن عندي ما تستعجلون به لقضي الأمر بيني وبينكم والله أعلم بالظالمين. وعنده مفاتح الغيب لا يعلمها إلا هو ويعلم ما في البر والبحر وما تسقط من ورقة إلا يعلمها ولا حبة في ظلمات الأرض ولا رطب ولا يابس إلا في كتاب مبين.
يطيب لنا أن نعيش الآن مع كلمة عميد كلية العلوم الطبية التطبيقية سعادة الدكتور ناصر بن علي الجار الله.
السلام عليكم. الحمد لله رب العالمين والصلاة والسلام على نبينا محمد وعلى آله وصحبه أجمعين. سعادة وكيل الجامعة للدراسات العليا والبحث العلمي دكتور محمد الشايع، أصحاب السعادة، عمداء الكليات، سعادة المتحدثين بروف أوليفر بروف ماركس ليفن، الدكتور بخيت الرشيدي، الضيوف الكرام، السلام عليكم ورحمة الله وبركاته. إن ما نشهده اليوم في المملكة من حراك واسع لإحداث التغيير الاقتصادي والثقافي الإيجابي نحو بيئة معرفية منتجة يأتي من حرص واهتمام خادم الحرمين الشريفين الملك عبد الله بن عبد العزيز حفظه الله لتطوير التعليم والتعلم، حيث أنه وجه بإنشاء عدة مراكز بحثية تخصصية في عدة جامعات ومؤسسات وطنية تعنى بتطبيقات تقنيات النانو، ليعطي دلالة واضحة على أن الاستثمار الحقيقي هو الاستثمار في العقول لتحقيق تنمية شاملة ومستدامة. إن عالمنا اليوم يتسارع فيه منتجات العقل البشري معرفة وتقنية، لذلك أدركنا أنه يجب علينا مواكبة مجالات العلوم المتطورة واستخدام التقنية في المجالات المختلفة، وتوجيه الاستثمار في مجالات المعرفة والمعرفة والبحث العلمي. إن التوجه العام لدينا في المملكة العربية السعودية هو دعم البرامج والأبحاث في تقنية النانو لتحقيق رؤى قائد مسيرتنا خادم الحرمين الشريفين الملك عبد الله بن عبد العزيز حفظه الله، للاستفادة من هذه التقنية لخدمة الوطن والمواطن والعالم أجمع. إنما يجعل الأمر أكثر أهمية أن الابتكارات بتقنيات النانو تعد الأكثر نمواً في العالم والأطول عمراً في الاستثمار. ومن منطلق حرص معالي مدير الجامعة الدكتور خالد بن سعد المقرن حفظه الله على تحقيق تطلعات ورؤى قادة هذه البلاد، فقد وجه بالانطلاق في هذا المجال وقدم الدعم اللازم، وتم تسجيل براءتي اختراع من إنتاج فريق بحثي من الكلية. الأولى استخدام نترات الفضة النانونية وضمها مع الخلايا الجذعية لتساعد في التئام الجروح في القدم السكرية. الثانية استخدام مركبات النانوية وضمها إلى الخلايا الجذعية لتساعد في تكوين الأوعية الدموية التي تغذي منطقة المخ بعد حدوث الجلطات الدماغية، ولعل الدكتور مارك سليفن أسهب في هذا وأجاد وأفاض. واستمرارا لتنفيذ توجيهات معالي مدير الجامعة حفظه الله بمتابعة المستجدات العلمية وخاصة في تقنية النانو، سعت كلية العلوم الطبية التطبيقية بجامعة المجمعة وباهتمام بالغ باتخاذ عدة خطوات كان من ضمنها إقامة هذه الورشة، ورشة عمل تقنيات النانو وتطبيقاتها في المجالات الطبية. وقد نتج ولله الحمد عن إقامة هذه الورشة عدة توصيات، كان منها ضرورة عقد تعاون وشراكة لخدمات مع المراكز البحثية العالمية والمتطورة في تقنيات النانو وفي المجالات الطبية. ولدينا مثالين، دكتور مارك ودكتور أوليفر، من الجهات الجيدة في هذا المجال. إنشاء وحدة النانو بالجامعة لتخدم التخصصات الطبية والعلمية والهندسية. أيضا دعم الأبحاث ذات الصلة بتقنيات النانو في المجالات الطبية وتشجيع الابتكار في هذا المجال بشكل أكبر. إضافة بعضا من المقررات أو ضمن المقررات الدراسية بما يختص بتقنيات النانو ومبادئها للتخصصات الصحية وغيرها من التخصصات العلمية والهندسية لتأهيل كوادر وطنية وتمكينهم من التعامل مع تقنيات النانو بأشكالها المختلفة. إنشاء أو التوصية بإنشاء بإنشاء كرسي أبحاث لتقنية النانو في المجالات الطبية أو في مجال محدد بعينه أو في استخدامات محددة أو في تطبيقات محددة لتقنيات النانو. إقامة ورش عمل مستمرة ودورات تدريبية في تقنيات النانو محليا ودوليا لتطوير أعضاء هيئة التدريس. ولا ننسى في هذا المجال أن كلية العلوم يفترض أن أو سيكون لها دور كبير جدا في هذا المجال، إذ أن العلوم الأساسية اللي تقف عليها تقنيات النانو وعلوم النانو وتقنياتها تقف على العلوم الأساسية في كلية العلوم مثل الكيمياء والفيزياء والرياضيات والعلوم الأخرى. ختاما أشكر كل من ساهم في نجاح هذه الورشة وفي نجاح هذا الحفل، ولا أريد أن أسمي حتى لا أنسى البعض وأترك البعض. أشكر الجميع صراحة، ونشكر أيضا شركة وادي السيليكا لدعمهم في هذه الورشة ومساعدتهم للكلية في إقامة هذه الورشة. أسأل الله العلي العظيم أن يعلمنا ما ينفعنا وأن ينفعنا بما علمنا، إنه جواد كريم، وأسأله تعالى أن يوفق الجميع لما يحبه ويرضاه. والسلام عليكم ورحمة الله وبركاته.
كلما زاد ارتفاع تحليقك زاد المنظر جمالا. هذا هو حال جامعتنا وكليتنا، كلية العلوم الطبية التطبيقية، من نجاح إلى نجاح، عاماً وراء عام، نعلو ونرتفع إلى آفاق سامقة لنرى بين الثريا. وفي طريقنا، طريق العلم والمعرفة، بدأ المشوار بجهود مؤسسية عملاقة وبروح يحدوها الأمل والثابة، فكان الجهد المبذول والسعي المتواصل لتترسخ عاماً بعد عام إنجازات ونجاحات سبقت بها جامعة المجمعة، وارتقت إلى سلم المعالي، وأصبحت نجماً يلمع في سماء العلوم والتقدم، ويرسم للحضارة خطاً جديداً، ويبني للوطن علماء ونوابغ لا يفترون بذلاً وبناء. فبارك الله هذا العقل الذي فكر، وتلك السواعد التي بذلت، وكلل جهود الجميع.
نسعى في جامعة المجمعة إلى أن يكون لنا بصمة علمية في شتى المجالات، والجامعة بفضل الله عز وجل حققت الكثير من الإنجازات العلمية، فقد نشرت أكثر من 380 بحث في كثير من المجلات العالمية، وقد حصلت على أكثر من 4 جوائز عالمية وبراءات اختراع، ومن آخرها كان في معرض جنيف الدولي لبراءات الاختراع. وها نحن في هذا اليوم نتطرق من خلال هذه الندوة إلى مجال جديد وحيوي في العلوم العلمية والطبية التطبيقية والهندسية، وهي تقنية النانو. ومن المعلوم بأن هذه التقنية تعتمد في معظم دراساتها على تطبيقات علم النانو من أجل التوصل إلى اختراعات واكتشافات علمية تسهم في النهاية في رفاهية الإنسان. وبفضل الله عز وجل في هذه الجامعة سوف نبذل قصارى جهدنا لأن يكون لنا عمل مميز في هذا المجال. نشكر الزملاء في كلية العلوم الطبية التطبيقية على هذا التنظيم، نسأل الله عز وجل أن يوفقنا وأن يسدد خطانا، وهذا والله أعلم، وصلى الله وسلم على نبينا محمد.
واصلت كلية العلوم الطبية والتطبيقية مشوارها الحافل المتنامي، فشهدت طفرات متلاحقة تبهر الناظرين وتنفع العالمين. فبدأت الكلية عامها 33 و34 بنجاحات وإنجازات عظيمة، فها هو نجم من نجوم الكلية لفت أنظار العالم وأبهر الجميع باختراعه الفريد والمفيد، فحصل به على الميدالية الذهبية في جنيف والكويت.
من خلال كلية العلوم الطبية التطبيقية وجدت البيئة المناسبة للتميز، وبفضل من الله قمنا بتحقيق جهاز قياس نسبة الهيموغلوبين من دم بدون أخذ عينة بمشاركة فريق البحث العلمي، وبحمد الله أخذنا وحصلنا على الميدالية الذهبية في مؤتمر جنيف الدولي، وعلى جائزة CII المهتمة بتطبيقات الحاسب الآلي، وحصلنا على الميدالية الذهبية في المؤتمر الدولي الرابع بالكويت.
هي بصمة تركها ليبعث بها إلى كل العالم: العلم ثم العلم ثم العلم. فهيا يا طلاب العلم جهدا وبذلا وعطاء لتصلوا بوطننا إلى المعالي. الريشة الذهبية جائزة تمناها الكثير، لكن الأسبق إليها والذي بذل قصارى جهده ليحققها أحد أبناء جامعتنا.
بفضل من الله ثم دعم كلية العلوم الطبية التطبيقية حققت نجاحات، ومنها المشاركة في المؤتمر العلمي الرابع، وحصلت على المركز الأول والريشة الذهبية، وذلك في ابتكار جهاز يعالج في تسطح قوس القدم، وحصلت على بعثة خارجية لإكمال الدراسات العليا، وأيضا تم ترشيحي من قبل وزارة التعليم العالي للمشاركة في مؤتمر موهبة.
وهكذا يكون النجاح تلو النجاح، ويأتي الإنجاز بعد الإنجاز، ليصل بنا إلى الطالبة سارة السلبود، ذات نجاحات نفعت بها البشرية جمعاء.
بسم الله الرحمن الرحيم. الوصول إلى القمة هو الأساس الذي استندت عليه جامعة المجمعة، وبفضل من الله ومن ثم دعم الجامعة ووالداي تمكنت من تحقيق بعض الإنجازات، ومن ذلك مشاركتي في المؤتمر العلمي الثالث والرابع والخامس لطلاب وطالبات التعليم العالي، وكذلك العمل مع فريق بحثي برئاسة الدكتور رائد البرادعي في اكتشاف علاج التئام جروح القدم السكرية باستخدام تقنية نانو الفضة، وبذلك حصلت على جائزة مدير الجامعة الدكتور خالد المقرن للتميز والجودة.
ويتوالى سجل الإنجازات والمفاخر التي يفخر بها طالب كلية العلوم الطبية التطبيقية، فقد أضيف إليه في ذلك العام استقبال معالي وزير التعليم العالي في مكتبه قيادات وطلاب الكلية وتهنئته لهم على ما تحقق في مؤتمر جنيف. إنشاء كرسي الشيخ عبد الله بن عبد المحسن التويجري لأبحاث الجلطات الدماغية. القيام بزيارة شركة الإلكترونيات المتقدمة ومناقشة سبل التعاون معها. عقد الملتقى العلمي الأول بالكلية. المشاركة في يوم البحث العلمي الأول للجامعة. حصول المبتعث للدكتوراه بكلية العلوم التطبيقية عبد العزيز الكثيري على جائزة التميز الأكاديمي للخريجين من أفضل 10 جامعات أمريكية. وهذا كله غيض من فيض. يدل على ذلك الإحصاءات التي زادت في كل قسم وفي كل زاوية من زوايا الكلية. فها هي كلية العلوم الطبية التطبيقية تمضي في طريقها العلمي، واضعة أمام ناظريها أهدافاً أسمى تريد أن تحققها لوطننا الغالي. فكانت تذلل كل الصعاب أمام طالب العلم ليصل إلى هدفه دون أن يعوقه في طريقه أي عائق. فكان الاهتمام بمعامل الكلية لتكون وسيلة متاحة أمام جميع الطلبة. أنجزت الكلية في ذلك تزايداً مضطرداً في عدد الأجهزة وزيادة الاعتماد على التقنية، إضافة إلى التدريب عليها والاهتمام برفع مستوى الطلبة، وكذلك الإداريين في الحاسب الآلي وغيره من الأجهزة المهمة في الكلية، بدءاً بكيفية التعامل معها، ووصولاً لكيفية تحقيق ما يصبو إليه المستخدم من خلالها.
تم بحمد الله تحقيق عدة جوائز بكلية العلوم الطبية التطبيقية محلية وإقليمية ودولية. وكما تم إنشاء كرسي الشيخ عبد الله بن عبد المحسن التويجري للجلطات الدماغية، تم تأسيس عدة معامل في أقسام المختبرات الطبية وفي مختبرات الأجهزة الطبية وفي مختبرات التمريض والعلاج الطبيعي والأشعة.
كما تم تأمين العديد من الأجهزة المهمة والتي تتيح للدكتور والباحث والطالب زيادة مستواه المعرفي والتطبيقي، ومنها جهاز الماسح الإلكتروني سيم، ويستخدم في دراسة السطح الخارجي للعينات بمختلف أنواعها وإظهار التكوينات الدقيقة لتلك السطوح. جهاز تسلسل الحمض النووي، ويستخدم لتحديد تسلسل الحمض النووي، والذي يعتبر المادة الأساسية لمعرفة الجين أو الجينات، والتي من خلالها يتم فهم تركيب ووظيفة الكائن الحي. وكذلك النوع الأكثر شيوعاً من المجاهر هو المجهر الضوئي، والفائدة منه هو إنتاج صورة مكبرة لعينة ما، وذلك لغرض دراستها وفحصها. مشوار من النجاح خاضته كلية العلوم الطبية التطبيقية جامعة المجمعة، سطرت خلاله بحروف من نور إنجازات رفعتها لتكون في مصاف الجامعات العالمية. بجهد مبارك متضافر بدأ من توجيهات معالي مدير الجامعة الدكتور خالد بن سعد المقرن، حيث كان الكل يواصل الليل بالنهار تفكيراً لما يمكنه من إيصال الصاعدين سلم العلم والعمل والإبداع. خطت الكلية في هذا العام تلك الخطوات التي نسأل الله جل وعلا أن يكتب بها النفع للبشرية جمعاء، ويسدد الخطى ويوفق الجميع لما فيه صلاح الدنيا والآخرة. كل الشكر لمن بذل لهذا الوطن الغالي ولرفعة أبنائه.
يطيب لنا أن نعيش الآن مع كلمة معالي مدير الجامعة معالي الدكتور خالد بن سعد المقرن، يلقيها نيابة عنه سعادة وكيل الجامعة للدراسات العليا والبحث العلمي الدكتور محمد بن عبد الله الشايع.
بسم الله الرحمن الرحيم، الحمد لله رب العالمين، والصلاة والسلام على أشرف الأنبياء والمرسلين نبينا محمد عليه أفضل الصلاة وأزكى التسليم. سعادة عميد كلية العلوم الطبية التطبيقية، أصحاب السعادة العمداء، وكلاء الكليات، ضيوفنا الكرام، أرحب بكم في بيت من بيوت العلم ألا وهو جامعة المجمعة. هذه الجامعة التي استعرضت قبل قليل عرضا مرئيا فقط عن أحد الكليات التي أبهرتنا حقيقة على الرغم من قصر عمرها الافتراضي بإنجازات علمية سابقنا فيها كليات لها أقدمية في هذا الوطن الغالي، وأيضا كليات أخرى في جامعات عريقة، كيف لا؟ وطلاب الكلية قد حصلوا على أكثر من 4 جوائز عالمية، لعل آخرها جائزة في معرض جنيف الدولي في دورته 41 العام الماضي. واليوم نسمع من خلال العرض المرئي وما ألقاه عميد الكلية عن أن هناك براءات اختراع تم تسجيلها في منظمات دولية. ما رأيته من خلال العرض المرئي على الرغم من انتمائي لهذه الجامعة منذ التأسيس إلا أنه فاق توقعي وفاق طموحي، ولذا أهنئ حقيقة طلاب هذه الكلية ومنسوبي هذه الكلية على هذه الإنجازات، وأبارك لهم القيادة بقيادة عميدها الدكتور ناصر الجار الله، وأيضا زملائي وكلاء الكلية وزملائي أعضاء التدريس، لأن هذا المجهود لا يظهر ولن يظهر بدون عمل وتفاني. مهما وصفت لكم مشاعر الحالية لن أستطيع أن أوفي حقكم، لكنها هي عبارة عن عبارات ألقيها لكم، أأمل أن توفي جزء من المجهود الذي بذلتموه خلال هذه السنوات. جامعة المجمعة سعت منذ نشأتها إلى أن يكون لها دور واضح وبارز في مجال البحث العلمي، ولقد أسست منذ البداية، كان من أول العمادات التي أطلقتها هي عمادة البحث العلمي، وذلك أيضا عبر إنشاء مجموعة من مراكز البحث المتخصصة في العلوم والهندسة، وأيضا الأقسام الطبية والقسم الآخر في الدراسات الإنسانية. دعمنا خلال هذا العام أكثر من 65 بحثا، منها 13 بحثا في المجال الصحي، ونطمح بإذن الله أن يكون لنا دعم أكبر خلال السنوات القادمة. بالنسبة فيما يتعلق بدعم أبنائنا الطلاب، صدر في خلال الأشهر الماضية موافقة مجلس التعليم العالي على مركز الابتكارات الطلابية. هذا الإنجاز بإذن الله سوف يترتب عليه إنجازات أخرى تحتضن أبناء هذه الكلية وأيضا أبناء الكليات الأخرى في الجامعة لمزيد من التفوق والإبداع في المجال الطبي والهندسي وغيرها من المجالات. حقيقة أنقل لكم اعتزاز معالي مدير الجامعة الذي كان حقيقة يرغب أن يكون متواجد وهو الراعي الرسمي لهذا الملتقى، إلا أن الظروف أحيانا تحول دون ذلك، ولولا الظرف الطارئ الذي حل بمعاليه لكان موجود بيننا، وهو يعلم يقينا ما تبذله هذه الكلية من مجهود عالي ومجهود رائع نتج عنه هذه الأعمال الإبداعية. لا أريد أن أطيل عليكم، لكن أشكر زملائي المتحدثين الذين ساروا هذه الساعات على الرغم من البعد الجغرافي من مكان هذه الجامعة، إلا أنهم أبوا أن يكونوا مشاركين معنا من خلال هذه الأوراق ومن خلال ال3 جلسات التي تم استعراضها في هذه الندوة. شكرا لكم جزيلا على وجودكم، شكرا على ما طرحتموه. أتوقع بأن الفائدة سوف تنعكس إيجابا في مجال تقنية النانو، وبإذن الله كمتحدثين سوف تروا الجامعة لها مجموعة من المبادرات العلمية والاختراعات في هذا المجال التي أنتم شركاء لنا فيها. أيضا أشكر زملائي وإخواني الحضور من أعضاء التدريس والطلاب وجميع الحضور دون استثناء على مشاركتهم وتواجدهم في هذه المناسبة. الشكر أيضا موصول لمعالي مدير الجامعة على رعايته ودعمه الكريم في كل مناسبات الجامعة. تقبلوا تحياتي، والسلام عليكم ورحمة الله وبركاته.
يتفضل الآن سعادة وكيل الجامعة للدراسات العليا والبحث العلمي الدكتور محمد بن عبد الله الشايع بتكريم من ساهم وشارك في إنجاح هذه الورشة، وهم الأستاذ الدكتور محمد بن عثمان الركبان، يستلمها نيابة عنه الدكتور رائد البرادعي، الأستاذ الدكتور أوليفر بوناود، الأستاذ الدكتور مارك سيلفن، الدكتور خالد بن محمد العبد الوهاب. الدكتور بخيت بن عوض الرشيدي. شركة وادي السيليكا لتقنية النانو. درع مقدم من شركة وادي السيليكا لسعادة عميد كلية العلوم الطبية التطبيقية الدكتور ناصر بن علي الجار الله. درع مقدم من شركة وادي السيليكا لمعالي مدير الجامعة الدكتور خالد بن سعد المقرن. وفي الختام لا يسعنا أحبتنا الكرام إلا أن ندعو الله لنا ولكم التوفيق، وها هو وقت الوداع حان وزمان البين آن، وإلى لقاء قريب وعلى الأفراح نجتمع، وسلاما على المرسلين والحمد لله رب العالمين.